Okay, now I talked about this before, but because Calliphoridae is so incredibly important in decomposition, in forensics, I'm going to go over Calliphoridae reproduction again. So this is going to be hopefully a bit of a review, but it is this reproduction that is the basis to everything when it comes to forensic entomology. So a little later on the semester, I'll give you a few cases and you're going to see it's just maggots all the time and how these things reproduce. So you're going to get used to me talking about this over and over and over again. So remember that Calliphoridae are holometabolous insects, just like all the Diptera. They have an egg stage, they go into 3 larval instars, they turn into the pupae, and then eventually they'll close into the adults. So it goes egg, larvae, pupae, adults, which makes them holometabolous. So last time I talked to you about the resource location of these Calliphoridae. They follow the scents upwind, they land on a potential resource, and they're going to assess the resource for food and for oviposition. So once the adult flies locate a suitable resource and find a suitable oviposition site, the gravid females will lay their eggs. So remember I told you about gravid. Gravid refers to females that have fully formed eggs that are ready to be oviposited. Okay, so these eggs are ready to go. If you dissect open these flies, you will see big old clutches of eggs. So they have about 200 to 300 eggs just hanging out in their abdomen, ready to go, ready to be oviposited. Now, in order to become gravid, remember that many of these females need to have a protein meal because they are anautogenous. Remember, autogenous means they can produce eggs without outside protein. They just use store protein. Autogenous, right? Autogenous or autogenous. So remember, genesis means coming from something. Auto means self. Autogenous and autogenous means they need an outside protein meal. So females can show up at a body, feed on the body, get that protein meal, wait a little bit, 24, 48 hours, however long it takes them, and then they will be gravid and ready to lay their eggs. So you're going to see different waves, basically, of these different types of flies. Some just coming in for feeding, and then they'll go off and find another body. Others are already gravid. They already have that protein. Now they're ready to lay their eggs. Those are the ones we care about when it comes to forensic entomology. Okay, so the female is going to lay her eggs by extending that telescoping segment in her abdomen, okay, that is going to extend to form an ovipositor. That ovipositor is very, very flexible. Remember, it's the ovipositor in the stinging insects that actually cause you to sting. Flies, while the females have an ovipositor, it's not super sclerotized, so it's not strong enough to sting you. So you don't get stung by flies, at least not by Calliphoridae. Okay, so they are going to use this very flexible ovipositor to lay eggs in these natural bodily openings and wounds. So the eggs are laid in those clutches, in bunches of eggs we call clutches. So here's a nice, beautiful clutch of eggs. You've seen this before. They're glued to each other. They're glued to the substrate with a thin layer of sticky substance that'll coat each egg individually. This becomes important because when you're trying to collect eggs, it's very hard just to collect a single egg. You won't necessarily see a single egg just sitting on something. If you do, something weird happened. Instead, you're going to see these large clutches of eggs, and when you pick it up, they're all going to come together. So it is hard not to damage the eggs if you try to pull them apart, so just don't. Just pick them up, keep them, and they will hatch out. So if you want to try to rear these flies, these maggots, don't try to pull apart the eggs. They're glued together. Okay, now this happens to keep these eggs protected. The outside eggs are the most vulnerable. These are the eggs that may dry out because of heat or because of really low humidity or something like that. These are the ones that are most likely to be eaten by predators. They're most likely to be parasitized. So those inner eggs are the ones that are more likely to survive. So this is just the way that this clutch works. So females can lay between 200 and maybe 300 eggs all at once. Okay, so they have the ability to lay a whole bunch of eggs all at once, and they can lay between 2,000 and 3,000 eggs over a 3-week period. So what they need to do is they will lay down their eggs, they'll lay down that clutch of eggs, they'll go off, get another protein meal because they're anautogenous. They will wait a little while, produce more eggs, become gravid again, go and lay their eggs again. So they'll do this 10 or 12 times, however many times it takes until they die. And their average lifespan is about 3 weeks. Now, the eggs can take, on average, 24 to 48 hours to hatch, although I have seen eggs hatch in as little as 9 hours. When it's really, really warm out, it's the perfect environment. They can go very, very quickly. So I say 24 to 48 hours on average, just because the U.S., North America, is very large, and we have very warm areas, very cold areas. All right, so this is the type of thing you want to keep in mind. Temperature is ridiculously important. So if you think about it just from this beginning, let's say I find a body that has eggs on it, I have to figure out what species those eggs are, how quickly they are going to hatch, and what the temperature is that is going to affect that hatching. If I find that this is one of those species that hatches super fast and we're in a super warm area, I mean, we're looking at just part of a day before these things hatch. If I'm more northwards, it's nice and cold, and this is one of those long gestating type of species, takes a long time. We're talking maybe 2 days before they hatch. So knowing a lot about these Calliphoridae is very important. Okay, now these eggs will hatch by breaking open these seams, so these weakened areas, and they hatch into their instars. So remember, Calliphoridae have 3 larval instars before pupation. And the maggots look like this. They are tapered down at the anterior end, all the way, they're much thicker at the posterior end, they're more blunt. At their anterior end, they have these pointed heads without a head capsule. Remember, this is because they are in Brachycera. The Nematosa are the ones with the big head capsule. Brachycera have no head capsule. Instead, they have this internal skeletal region and these mouth hooks that attach to this internal skeleton. So these mouth hooks will allow them to feed. They will extend the mouth hooks and scrape in the environment. They will eject digestive juices in order to do exodigestion, that sort of thing. Now, the posterior surface has the sphericals. Remember the sphericals? Here's a single spherical. It has these little spherocular openings that will open and close with musculature to allow oxygen to diffuse in and out. So there are 2 sphericals with a varying number of spherocular openings in each spherical. And those spherocular openings vary with the instar. Okay, so remember that the instars are based on how many times they've molted. So the first larval instar is very tiny. It is about the size of the egg, and they're so small that they can, they're pretty in danger right there. They can dry out very easily. They can be eaten very easily. They can't move very far because they're so small. They're going to feed right near the egg mass, or where that egg mass was. And they're going to go through this first instar pretty quick because it's pretty dangerous to be there. You can tell first instar because, one, they're super, super tiny, and they have a single U-shaped spherocular slit. They will then molt into the second larval instar. They will move into less putrid material. They'll do exodigestion. They'll start feeding. And they have 2 spherocular slits. They'll then molt into the third larval instar. And the third larval instar is really the voracious feeding instar. Okay, so they will range from 8 to 23 millimeters in length when they're in that third instar. Usually they're white or cream colored, although some that you'll see in a little while are kind of spiky and they're really sclerotized, so they're much darker in coloration. But the terminal segment, that last segment of that third instar, has these tubercles, these cone-shaped tubercles around the perimeter. They've got the sphericals, and they have 3 spherocular slits in their sphericals. Now, the Calliphoridae spherocular slits look very different from some of the other families. So in the Muscidae or in the Sarcophagidae, say, they're going to have different shapes. Muscidae, they're going to have these little sinuous or these S-shaped spherocular openings. In Sarcophagidae, they have these slits like this, but instead of pointing sort of downwards and towards the midline like you see here, they will point straight up and down, or they're pointing in a different direction. So it's pretty easy to tell the family just from these spherocular slits. So the Calliphoridae will have these slits that point at an angle towards the midline or towards the center of that posterior area. And they tend to be straight, these foracular slits. So they're not S-shaped, they're not sinuous, and they're not pointing straight up and down. They're at an angle. That's the easiest way to tell Calliphoridae from pretty much any other larvae out there. You'll also see that the sphericals are sitting flush with the surface of this posterior segment. So they're not in these very, very deep pits. They're not sunken into that last segment. In a couple of weeks, I'll show you some that are, which is the different family. So just a few rules of thumb in order to tell Calliphoridae from every other family out there. Okay, so it takes between 4 and 21 days on average for the larvae to go through their full 3 larval instars. Okay, so this again depends on the species. It depends on the temperature. The warmer it is, the faster they're going to go through these things. The colder it is, the slower they are. So just keep that in mind. Now, so these naturally warmer areas, they're going to go really, really quick. In fact, I've seen some of those eggs that hatched within 9 hours, they can get to the 3rd instar within 24 hours. So it'll be no maggots on one day. The next day we've got 3rd instar maggots. So they can go really fast under the right circumstances. Now, because they are all laid in these big clutches, 200 to 300 eggs all at once, you will also see that there's a whole bunch of other adults that will come and lay their eggs one on top of the other. So you tend to see thousands of maggots all at once. We don't see lone maggots. This is because of maggot mass feeding. So when maggots feed, they have exodigestion. They will all basically puke up their digestive juices, put it into the environment, and it's like an external stomach in that case. They're going to break down that external environment or that meat, and then they're going to scrape it up with their little adorable mouth hooks. Now, think about that then. A single maggot trying to just puke up its own teensy tiny bits of digestive juices is not going to be able to feed very well. It's not going to be as efficient because there's just not enough of the enzymes out there in order to break down this protein efficiently. They can get a little bit, but not a lot. So that becomes an issue. Now, if you have thousands of maggots, though, they're all puking up a teensy tiny bit of enzyme, but there's going to be so much enzyme in the environment then that you're going to get a massive amount of digestion. So the more maggots that are there in this maggot mass, the more efficient the feeding is going to be. Basically, the bigger the external stomach is going to be. This is why we see these maggots feeding en masse. So in a lot of other disciplines, we talk about competition and there's a maximum number where we tend to see everything leveling off. The maximum number for maggots is super, super high. In fact, we really don't see maggots in this first wave of decomposition going into any type of real competition, just because it is so much better for them to be in a group than it is to be alone. So we have these competing rules of ecology. So instead of competition, they tend to work in concert. Neat. Okay, so they're going to feed like this, and they're going to feed, and this is why we see some really fast decomposition in bodies, and these bodies can be eaten completely by maggots very, very quickly. Once the maggots have finished feeding, once they've gone through all of that, they've gotten all their calories, remember that's their whole job as a maggot, gotten all these calories, they will move into the dispersal phase. We also call these the wandering maggots. So once they've finished feeding, they're going to disperse from the body and find a protected area in which to pupate. So they wander away from the body. And although wandering makes it kind of sound like, oh, they're aimless. They're really not. They tend to go in mass. They tend to go in a group because they're all about the same age. They're all in the same life cycle right there. And they are really protected in large groups. But they're going to move away all at once in sort of these big roiling masses. You can see every once in a while you'll see them dispersing out in this big starburst pattern in all directions from the bodies. A few just go off on their own, but they're basically going away to find a nice protected place. In areas where the body is found on soil, they will dig down into the soil a few centimeters or so just to be nice and protected. If the soil is too rough for them to dig down, or it's a really, really rocky thing, they'll just go under stuff. They'll go under under leaf litter, under rocks. Sometimes they go under the body directly and pupate there. It's basically they're trying to find a good place in order to break down their entire internal system and build it back up into the fly. Now, this stage then is often called the pre-pupal stage. So it is majorly characterized by the maggots discontinuing feeding. This is one way where we can tell these dispersing maggots, these pre-pupal maggots from any other stage, is these younger maggots are going to be actively feeding. And if you look at them on their abdominal region, you can see their internal system. So you see this dark line that's right here? That's their alimentary canal. Okay, remember way back to internal anatomy. So you can see their crop. You can see their foregut. And if they're actively feeding, there's going to be a very dark spot right there on the foregut. Okay, once they've turned into that wandering stage, they're going to clear out that alimentary canal. Okay, so they're not going to feed anymore. You won't see anything dark on the foregut. So it'll be kind of like up here. See how these are just bright white? There'll be nothing in there. That is a big indication that they've moved into that pre-pupal stage. They're no longer feeding. Now they're looking for a place to pupate. Now they're going to disperse somewhere between 15 and 20 feet away from the carrion. And this depends on the environment. So in normal outside environments like grass or just soil, 15 to 20 feet, they're going to dig into the soil, or they're going to crawl under leaves, rocks. If they're indoors, they're going to crawl under carpets, behind curtains, under furniture, that sort of thing. On smooth surfaces, they can move up to 100 feet away from the feeding source. So this can give them a great place in order to hide. And so you want to start looking for these pre-pupal and these pupating maggots a little further away from the body, because you want to find the oldest maggots. And we'll talk about that a little bit more when I get into actually how to do time of colonization estimation. Is the older the maggot, the more accurate your time of colonization. If you don't find these ones that have actually left the body, you're going to be underestimating the time of colonization. Now, the dispersal of maggots is also important because it could seem like there are more maggots on a body than there really are, because they're all moving away. So there is a case that a friend of mine did, and she was the forensic entomologist. She was specializing in animals at the time. And so she was called in to a necropsy. So necropsy is when an animal is basically given an autopsy. So autopsy is in humans, necropsy is in animals. So when you have a pathologist, an animal pathologist that is trying to figure out what killed an animal. So there was a case where my entomology friend was called in. A woman came in with her dog, and the dog had an open wound. This happens really, really commonly, especially in long-haired dogs. You'll see that they get injured or something like that. The wound starts to fester. You don't notice it because the hair is covering it. Flies will notice it, though, because they have that ability. They'll come in, lay their eggs, you get maggots on that wound. A lot of times, this actually doesn't cause any issue because these maggots will feed on the decomposing material. It doesn't cause any pain. It actually helps the animal stay alive because it's getting rid of all that really nasty tissue, and that wound will close up pretty quickly. Anyhow, the owner notices that there's some maggots on this dog, freaks out, as owners are wont to do, brings this dog into the vet. The vet sees a whole bunch of maggots just crawling all throughout that dog's body, like all throughout the fur, says, okay, this dog is just way too far gone, covered in maggots. They put the dog down. During necropsy, they found that the dog was perfectly healthy except for a very small wound on its back, and that wound was clean. Okay. So the entomologist was called in because the pathologist couldn't figure out why the dog was dead. The entomologist was looking at it, and it turns out that the maggots that the vet saw were in the wandering stage. They were pre-pupal maggots. So these maggots had been feeding on this little tiny wound, cleaned it out completely. They were now ready to pupate, and they were trying to leave that dog. This is actually when a lot of owners notice maggots on their animals because they're moving through the fur. Otherwise, they're going to be in that wound. They're going to be hiding. They're going to be feeding head down, butt up, breathing, doing that exodigestion stuff. They're not going to be noticed until they try to leave. So if that vet had understood this basic life cycle of the maggots, then they could have said, oh, these maggots are no longer feeding. Let's clean up this dog, shave it, try to find the wound. They would have found the wound. It was nice and clean. They probably could have put him on maybe a few more antibiotics, but at this point wouldn't have needed it. Would have just dressed that wound and that animal would have been fine. Because they didn't know this life cycle, they thought that this animal was covered in maggots. It wasn't. These are just really big maggots trying to leave that body. So this was a super, super unfortunate situation. This animal died for no reason. It had gotten a simple wound. It wasn't being neglected. And the vet just didn't know what they were talking about. So I always tell this story because it just drives me nuts. You need to know the difference, especially if you're working with animals, between maggots that are actively feeding and maggots that are just trying to get away and pupate. All right. Now, they turn into this pupal stage, so they're going to find this protected area. So once they're in that protected area, that pre-pupal stage, that wandering maggot is going to shorten, and instead of shedding its exoskeleton, it's going to harden the exoskeleton into a pupal casing. So you'll see that the first couple of minutes of the pupal stage look a heck of a lot like a maggot. They're just a little bit shorter, and then they start to harden their exoskeleton. So really new pupae tend to be much lighter in coloration than the older pupae. However, when we look at color and stuff, it's really hard to tell the difference, especially with a whole bunch of like decomp fluid and stuff going around. But in general, you can look and say this lighter pupae right here, that's a little bit younger than this older pupae, this darker pupae right there. Okay, so that's just sort of another rule of thumb. Now, the pupal stage can last between 3 to over 20 days, depending on the species and the temperature, and their adults have been known to eclose from pupae that are many months old. Now, as the pupae go through this pupal stage, what they're doing is they are breaking down their internal anatomy and going from this maggot and using a bunch of stem cells and imagonal discs and things of that nature to rebuild their entire body. So the outside of this pupal casing will still have some of the characteristics of the maggot. You can still see the sphoracular openings, you can still see the mouth hooks, but all the other stuff inside is going to be broken down by enzymes and rebuilt. So this is what it looks like. So this is a series of pupae where the investigators took the actual puparium, that pupal casing, off so we could see what was happening. So this happened early on. Still looks like the maggot here, but then you start to see the adult mouth parts and the antennae form. You start to see the wings form. Gets bigger and bigger. Oh, look at that. You can really see the wings there. Okay. So they just start forming more and more, and then you start seeing the compound eyes, you start seeing the mouth parts, they start to get some coloration, until right before they're ready to eclose from the pupal casing, you can actually see their full body, all ready to go. So if you were to dissect open a really old pupae, one that's just about ready to eclose, you could actually identify the fly inside. If you were to look at the outside of the pupae, you might be able to identify what that fly is based on these old larval characteristics that are left behind. So there's a lot you can actually tell from these pupal casings. Now here's this video again. Here's our pupae. Notice that there's no feeding, so it's in that pre-pupal stage. Here it is shortening, hardening that exoskeleton. You can see that the mouth hooks are left behind. You would see the sphoracular openings on the posterior end. Look how dark these pupae are. They're older. Remember, they use that selenium to pop open the end of that pupal casing. There it is. Hi. See those little aerostate antennae? Hey, notice they're plumose Calliphoridae. Okay, so coming on out, hey, look at that selenium coming out between those compound eyes, and it's going to wiggle its way out, and then it's going to take a few minutes to fully sclerotize. Depending on species, it can take different time. I've worked with some species that take about 45 minutes on average to fully sclerotize. So it's wiggling its way out. See how delicate it looks right here? This is very dangerous for this fly because things can eat it really easily. What is lucky for them, they can run like mad at this point, and they will run. It's really hard to catch them. Finally, just a quick reminder, it's very easy to tell the difference between male and female Calliphoridae. The males have these large eyes that are very, very close together. They touch at the surface or the apex, while the females have really wide spacing between those eyes. All right, so that's the basics of the reproduction again. Let me know if you have any questions.